Hello, ladies and gentlemen. Today I'm going to start talking about Chapter 3, titled Cellular Form and Function. You might notice that since I discussed Chapter 2, I have gotten older, more gray hair, but I did get a haircut. Okay, Chapters 3 and 4 are a short course in cellular biology. In Chapter 5, we will finally get into tissue types and then go on from there into organs and organ systems. There are 4 sections in Chapter 3. Let me break. It's a really good idea right now, before we get any further, to print out the study guide that's on page 109 of your text. These are excellent tools for your study for the unit exams. Okay, the 4 sections in Chapter 3 include concepts of cellular structure, the cell surface, membrane transport, and the cell interior. Now, I am certain that all of you have heard of cells. Every single live organism, from a one-celled bacteria on up, including humans, are composed of cells. One of the main differences, of course, is the number of cells and how specialized they are. On Figure 3.1 on page 79 shows the different forms or shapes that a cell in the human body can take. Each different cell type, cell shape, has a rather specific place where it's located and reason for this particular shape. I want you to at least have heard of these. Some of them you will hear much more about as we go on into the chapter. Page 80 starts a discussion of the basic components of a cell. Every cell in the human body has the same basic components. Around the cell is what's called a plasma or cell membrane. This is a very specialized membrane that we'll talk about just a little bit later. Inside of this membrane is a liquid type, gelatinous type fluid called the cytoplasm. And it also contains, if we look at a picture later on, or if we had an electron microscope, you could see many different specialized parts in this cytoplasm. Okay, Figure 3.5 on page 82. I want you to put a little marker there if you had a textbook, or just remember page 82. This is a color structure of a representative cell. Everything that we're going to discuss, all the different parts of a cell, are shown in this picture. And it is a good idea to have a picture to refer back to. Even in my days of engineering or math, I liked to draw pictures because it made things more clear to me. First, let's talk about the plasma or the cell membrane. This membrane is very specialized and does go all the way around the cell. It holds the cytoplasm in, if you will. The cell membrane or the plasma membrane is let me back up. The cell membrane, 98% of it is lipids, are made of lipids. and 75% of these lipids are phospholipids. Figure 3.6 on page 83 shows a very good schematic of this cell membrane. The phospholipids are lined up such that the amphipathic, these are scrub that word, part of this molecule, the phospholipid molecule, likes to be in water. This is the hydrophilic phosphate-containing heads, which are to the outside and the very inside of the plasma membrane. The hydrophobic tails of the phospholipid are in the center, and they avoid contact with water. So the cell membrane, if you are asked, is a phospholipid bilayer. It's made up of two layers, mostly of phospholipids. Now, look at the other structures that are also found in the plasma membrane. These are very, very important. These are called membrane proteins. Some of them are integral proteins in that they penetrate inside the bilayer, or sometimes go all the way through it. Those that go all the way from the outside to the inside are called transmembrane proteins. Some are peripheral proteins and do not protrude into this phospholipid. Now, the purpose of these proteins include several different things listed on pages 84 and 85. They are receptors that join on to other things that are floating around in the blood or the other liquid in the body. Look at figure 3.8. This shows you a schematic of the 6 different functions of these plasma membranes. And for your information, they are not the same color as they are shown in this textbook. I've never been able to look at a cell through an electron microscope to see what exact colors they are made up of. But the different functions include receptors, second messenger systems, enzymes, channel proteins, carriers, and cell identity markers. Oops, there's a 7th one. Cell adhesion molecules. One of the these all are important, but one that your author wanted to spend more time on right now is called a second messenger function. And that is pictured better on figure 3.9. If you follow through that, see the small numbers. There is a receptor to the outside of this transmembrane protein. A messenger such as epinephrine or some other neurotransmitter binds to this receptor in the plasma membrane. This is the signal then for some protein, here called a G protein, to be released. And then that G protein on the inside of the cell now, in the cytoplasm, can be another signal for other things to occur. And the G protein is called the second messenger. The first is the epinephrine, and the second would be the G protein. Okay, skipping along, we're going to skip about microvilli, cilia, etc. You'd have to know more about that if you go into cellular biology as your career. Section 3.3, membrane transport. It is very important that you have a very basic understanding of how various chemicals, molecules, ions, atoms can pass through the membrane from the outside to the inside of the cell and vice versa. This plasma membrane can be both a barrier and a gateway. It is called selectively permeable in that it allows certain things through, such as nutrients, wastes, gases, but other things are prevented from passing through the membrane by themselves, such as proteins or phosphates. There are several different ways that this membrane transport can take place. Filtration, simple diffusion, osmosis, and then carrier-mediated transport, and finally active transport. Filtration is a very simple process with the force behind it is strictly a physical force. For example, in the bloodstream inside a nephron in the kidney, certain products can be forced by the blood pressure right through the membrane into the inside of the nephron. Simple filtration. Water, salts, nutrients, and other solutes can be cross the membrane in this way. Also, there is simple diffusion. Simple diffusion is the net movement of particles from a place of higher concentration to a place of lower concentration of those particles. They're able to do this because each of these particles is moving and it has kinetic energy. Now, here's a term that I want you to understand. Simple diffusion means that no energy is being used and the particle is going down its concentration gradient. That is just a way of saying it's moving from a place of higher concentration to a place of lower concentration. Again, does not require the use of any energy. How fast can it go? That's determined by several different things. And I want you again just to at least have heard these terms and understand a little bit about how they can make the diffusion go faster or slower. One is temperature. The higher the temperature, the faster the diffusion. The molecular weight of the product. The lower the molecular weight, the simpler it can diffuse. The steepness, in quotes, of this concentration gradient. That just means the difference between the highest and the lowest concentration. If there is a large difference in concentration, then the diffusion can take place faster. Membrane surface area. The larger the surface area, the more particles that can diffuse in a specified amount of time. And then finally, membrane permeability. Different membranes in different cells are permeable to different kinds of products. Another thing very closely related to diffusion is osmosis, and you have probably heard that term. Osmosis, like simple diffusion, does not require the use or expenditure of any energy. The difference is, in osmosis, we are looking strictly at the net flow of water from one side of a selectively permeable membrane to the other. Okay? Diffusion is a movement of the particles or the gases. Osmosis is a movement of water. It too is moving down its concentration gradient. For example, if we have one side of the membrane that is hypotonic, means less than the normal tonicity of fluid, that means there is more water than there are solutes. So the water is at a higher concentration. A hypertonic solution has less water than the hypotonic. So there is less water on one side, more water on the other. The water will tend to pass from its higher concentration to its lower concentration. Some of the water is able to go right through the phospholipid bilayer, but it passes much easier through pores or transmembrane proteins called aquaporins. Aqua meaning water, porin meaning like a small pore. These are proteins specialized for the passage of water. I want you to know a little bit about osmolarity and tonicity. Osmolarity, for those of you who have not studied chemistry, is the number of osmoles, chemical term, of a solute, such as sodium, chloride, potassium, etc., glucose, per kilogram of water. Okay? It is the ratio of the number of particles to the amount of water. Tonicity of a solution we've already discussed when we said hypotonic has a lower concentration of the non-permeating solutes. Hypertonic has a higher concentration. If we have an isotonic solution, then the concentration of the solvent and solute on both sides of the membrane is the same. Look very quickly at figure 3.16. Picture B shows a red blood cell, which, by the way, has no nucleus, and it has a concave surface on both sides. If this is put in a hypotonic medium, the water will flow into the cell because the water has a higher concentration outside of the cell. Because of this, then the cell can actually swell, such as shown in picture A. Perhaps if the swelling is enough, it can even rupture. Now, if this solution is hypertonic, then this cell can actually shrink, such as in picture C. Okay? So the tonicity, hypertonic, hypotonic, or isotonic, between the two sides is very important. Now let's talk about carrier-mediated transport. We're going to discuss three different processes classified altogether as carrier-mediated transport. That means there is some physical carrier that allows certain particles to be transported across a membrane, into or out of the cell. Okay. Now, look at figure 3.18 3.19, and 3.20. These are examples of the three different kinds of mediated transport. 3.18 shows what is called facilitated transport. There is a great difference in the concentration, for example, of sodium between the outside and the inside of a cell. Likewise, there's a great difference between potassium from the inside to the outside. Some of these ions will leak across, just magically pass through the membrane. But the body wants to keep the concentration as close to normal as possible. And this facilitated transport mechanism is able to do that. You see one particle, a little red sphere from the extracellular fluid going into the transport protein. This, when it binds inside the protein, then the protein changes shape and the ion is released to the inside. Facilitated diffusion. Okay, then look at the two pictures I also showed you. These are primary active transports. These do require energy in order to function, okay? We talked about simple diffusion and osmosis. No energy is required. But in active transport, we do require the use of energy. And where does this energy come from? It comes from the energy that is stored in the ATP or an ADP molecule. When that is chemically changed, that molecule, it goes from adenosine triphosphate to adenosine diphosphate plus a free proton with the release of energy. And then that energy can be used many different places. In secondary active transport, you will see glucose and sodium crossing the membrane. This is secondary active transport. Much more commonly, you will hear, and I want you to understand figure 3.20, the sodium-potassium pump. The sodium-potassium pump. I was hinting at that when I talked about facilitated diffusion. In this picture, the red spheres are sodium, the bluish colored spheres are potassium. And you see a purple transmembrane protein, and this is called the sodium-potassium pump. Also called in parentheses the sodium-potassium ATPase. The ATPase is an enzyme that will break apart the ATP. So you see in this particular pump, 3 sodiums are binding to the inside of the transport protein, 2 potassiums are binding to the inside of the protein, and then when energy is released by the hydrolysis of ATP, this membrane changes shape, and the sodiums are released to the inside of the cell, and the potassiums are released to the outside of the cell. This is the means by which our body can maintain the normal concentration of these very, very important ions in the body. Okay. Now, another type of transport is vesicular transport, a vesicle. A vesicle is a small particle, a small membrane-surrounded particle that can be released from the inside of the cell to the outside. These vesicular transport processes can move large particles, droplets of fluid, and many other different types of molecules through the cell membrane itself. Look at figure 3.21 and follow it from number 1 all the way around to to figure number 7. A phagocytosis cell, a phagocytic cell, is one that encounters a particle of foreign matter, and this particle is recognized as being foreign. This particular phagocytic cell opens up, swallows, if you will, this foreign particle, breaks it down, and that phagosome fuses with the lysosome that we'll discuss here in a minute, and then that is packaged in a small vesicle, which is then indigestible and is removed from the body or from the cell in a process called exocytosis. Endocytosis means into the cell. Exocytosis means out of the cell. Okay? Now, table 3.3 lists all of the different methods of membrane transport. No, you do not need to memorize all of these, but you should understand what the names mean. All of them are shown in with very good diagrams in other places in this chapter, as I pointed out. Okay, finally, figure 3.4. The cell interior. Figure 3.25 on page 100 shows the cell interior, but this picture is just much too busy. I suggest going back, and if you can, print off figure 3.5, which is back on page 82. First, we'll discuss very briefly the cytoskeleton. Just like our own skeleton is made up of bone and cartilage. The cytoskeleton is a network of protein filaments and cylinders that structurally support the cell, determine its shape, organize its interior, and can contribute to the movement of a cell if it is moving. Okay, now we'll go on into the cell, looking at figure 3.5. An organelle. An organelle is a general term for something that is inside the cell that can carry out a very specialized metabolic task. Some of these organelles excuse me, are surrounded by membranes, others are not. Once again, folks, you don't have to memorize all of this unless you are going on forward to get your PhD in cellular biology. First, the nucleus. If you look at figure 3.27, On the left is a light microscope picture. This is one that you, in a simple laboratory, you could see in a microscope. You would see the dark staining nucleus. This is the largest organelle in the cell and is usually the only one that you can really see through this light microscope. It is very, very important. It contains all of the cell's chromosomes. Therefore, it is the genetic control center of all other cellular activity. Now, most cells have a single nucleus. There are just a few exceptions. One, as I mentioned, is the red blood cell. They do not have any nucleus in their mature form. Now, others have more than one nucleus. Examples, real good example, is the skeletal muscle cell that we will look at in another chapter. The nucleus is surrounded itself by a nuclear envelope. And this is another phospholipid membrane, just like the cell membrane itself. This nuclear envelope is perforated in many, many different places by what are called nuclear pores. And these allow certain things to pass into and out of the nucleus, mostly out of. We will see the importance of these again later. When the DNA is copied by RNA, goes outside and then forms proteins. Okay, endoplasmic reticulum. Good word. Pair of words, rather. It means, in Latin, wish I had some water, a little network within the cytoplasm. There are two different types of these ER, endoplasmic reticulum, that you could see through an electron microscope. Some of it is rough. Look at figure 3.28, page 103. You can see the little red beads, if you will. This is rough ER. Those little beads are ribosomes, very small granules of protein. To the right in that picture is smooth endoplasmic reticulum. And in that part, there are no ribosomes attached. The endoplasmic reticulum has a couple of primary functions. One is that it synthesizes the naturally occurring steroids in a body and other lipids. It also detoxifies alcohol and other drugs and manufactures all of the membranes of the cell. We've talked about all the different places where you might see these membranes. This all occurs in the endoplasmic reticulum. Next are the ribosomes, those little red beads that you can see, some of which are attached to the endoplasmic reticulum, and some are free to float around in the cytoplasm. These ribosomes are small granules of protein and RNA. We'll learn more about that in chapter 4. RNA. DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. They have very important different functions that we'll study next chapter. Okay, so the ribosomes are made up of protein and RNA. This RNA is very, very important because it aids in the coding of the genetic messages that are formed in the nucleus of the cell. This RNA in the form of messenger RNA is able to move through the nuclear pores we talked about a minute ago, and then guide in the assembling of amino acids in very specific patterns to form proteins. Our figure 3.29 called the Golgi complex. This is a small system of cisternae that synthesizes carbohydrates and also puts the finishing touches on protein and glycoprotein synthesis. I like the way that they use those terms, finishing touches. I like that. Now, one of its most important products are the Golgi vesicles. These are able to include various products inside the vesicle and can move it either just around the cell, or it can actually take it to the outer cell membrane and release it to the outside. Next are lysosomes. These lysosomes are shown in figure 3.30, and these basically are packages of enzymes that are bounded by a membrane. We don't want these enzymes to be active all the time, only when they're needed. So the lysosome is able to keep them packaged until they are needed. A peroxisome is very similar to a lysosome, but it has different enzymes, and it's produced by the endoplasmic reticulum rather than the Golgi complex. Mitochondria. Mitochondria are shown very well in figure 3.30a. These are very important because they are specialized for synthesizing ATP, our energy molecule. Okay? And that, folks, is the end of chapter 3. Hopefully you're getting some use out of these. I have had a few messages stating that you appreciate these, and please keep them coming, somebody said. And I intend to do that. I think everybody could get a little bit of use out of watching this if you can have a free 20, 30, or 45 minutes. I'm concluding this now, and this one's only 38 minutes long. So this you should be able to watch very easily. Ciao and goodbye.